for about 12 days. And then we should point out earlier that here, if we just a single knockout of the CDK2 or CDK6, the mice are okay, they develop okay. They come out, they become just like a normal mice. But if the scientists do the double knockout, CDK4 and CDK2 at the same time, they die about after birth. Okay? P means they just after birth, the mice die. Okay? If you do the CDK4, CDK6 knockout, the embryo could not develop beyond 16.5 days. The scientists even go extra mile, went extra mile to show the triple knockout. As you can see from the triple knockout here, CDK2, CDK4, and CDK6 triple knockout. The embryo died at 12.5 days. So you got a feeling about all these different cyclic CDK. They play important role during developmental process. And the most important one is the CDC2 one. Okay? The other one play important role, but they sometimes their biological function could be compensated by other CDK. So scientists have done the CDK2 knockout. They also done the cyclin knockout as well. And now you can see, okay? Cyclin A knockout. I don't know why this is kind of got cut off. Do we have an adjustment here? Yeah, we missed this part. Is that? My screen was okay, but anyway. Huh? This side is okay. Okay, that side. So, but you do have a handout, right? Everybody have a handout right now? Okay, so maybe you could bear with that, okay?
He's already checked.
Huh?
He's already tried to fix it.
Okay, that's fine. Everybody have handout, and then you know what I'm trying to point to, okay? So the cyclin A knockout is viable, okay? The only thing lethal again here, some of the knockout mice, cyclin B1 is lethal, okay? Again, the cyclin B1 is involved in mitosis. So you don't need to memorize this, but I just want to share this with you so you can get a sense about these different cycling CDK play important role in cell cycle. So when the scientists knock them out, they have different phenotypes. And some of the phenotypes tell us how critical it is for the developmental process. Now, after finish the positive regulator of cell cycle, now I want to move into the next step to talk about the negative regulator of the cell cycle machinery, or the so-called brake system of the cell cycle machinery. Before we go on doing that, the cell cycle machinery is very complicated because biochemically, we have 4 different ways to stop the cell cycle machinery. But for us to ride on a motorcycle or bicycle, we easily to stop our motorcycle or bicycle, or even car, right? You just step on the brake, the car or your bicycle will stop. But in cell cycle machinery, we have very complicated brake system to stop the cell cycle machinery. The first one, as you could tell here, the cyclin CDK complex. This is cyclin CDK complex. They are in an active form, but if they bind with another protein called CKI, stands for cyclin-dependent kinase inhibitor, the whole complex becomes activated. Okay? So that's one mechanism to kill the cell cycle machinery. The second way to stop the cell cycle machinery is by dissociating the cycling away from the whole cycling CDK complex. In this case, cycling could be degraded, and then these CDK become inactivated because CDK got a name. Without cycling, this kind of CDK is no longer activated. So that's the reason the CDK has a name called cycling-dependent kinase. Okay? The third way to stop the cycling CDK complex is by a phosphatase. Okay? This particular phosphatase called CAP, CDK-associated phosphatase. The CAP will do is to dephosphorate on particular residue on CDK called serine 160. Once this particular phosphorylation site got dephosphorated, this cycling CDK becomes inactivated. The number 4 mechanism or the brake system to stop the cell cycle is by associating with a group of kinase called V1-MYC1 kinase. This particular kinase will add inhibitory phosphate group on the 2 important residues, serine 14 or tyrosine 15. By adding a phosphate on this particular residue, the whole cycling CDK got inactivated. Okay? So our cell cycle machinery even come out with such a delicate system to stop the cell cycle machinery. By binding a CKI, by dissociating cycling, by associating with the phosphatase, or being phosphorated by inhibitory kinase. So this is a negative regulator of cell cycle. So we will focus on the first part of by associating with the CKI. CKI stands for cycling-dependent kinase. CKI basically has 2 major families. When you read the literature, sometimes when people talk about cell cycle regulation, they will tell you, oh, which CKI has been upregulated, which CKI has been downregulated. There are 2 families of the CKI, called IN4 families or CKIP family. IN4 family stand for inhibitor of CDK4. IN4 stands for inhibitor of CDK4. They are including P16, P15, P18, and P19. This stands for IN4A, B, C, D. Okay? IN4A and IN4B, I will call them a very important tumor suppressor gene because these important CKI, they are mutated in many types of cancer, including familial melanoma. Okay? The other one called CKIP family stands for CDK inhibitor protein, and that including P21, P27, or P57. And these are important CKI have their own target. Okay, for example, IN4 always target on CDK4 or CDK6. As for P21, P27, they do both. They target on cycling CDK, the G1, early G1 cycling CDK. Also target on late G1 cycling CDK, called cycling E-CDK2. Okay? As you can see from this cell cycle clock here, G1, S, G2, N is the shortest of the whole cell cycle phase. Okay? And each phase is controlled by cycling CDK or CKI inhibitor. Okay? When the cell tells the cell cycle to move ahead, usually cycling CDK inhibitors, like CKI, they will become inactivated. So we can spend a little bit time about these CKI. CKI, as I mentioned earlier, stand for CDK inhibitor. Okay, 2 families. IN4 family here. The reason They call info family because they are structurally related, as you could tell. They all have this anchoring repeat there, okay? And they've been induced by quite a few different inducers, okay? For example, one of the inducers for P15 is called TGF beta, transforming growth factor beta. This is a very interesting cytokine. Sometimes they will stop the cell from cell proliferation. And this particular cytokine also involved in metastasis at a late stage of the cancer formation. Okay? Another group, which we're going to talk about quite a lot, is called CCSTI family. And they have P21, P27, or P57. Again, they are structurally related because at the very end terminal, they have this CDK binding motif there. Okay? Again, this group of CDK inhibitors are induced by various signals. For example, the most important one and critical one is induced by P53, okay? Everybody should know about P53 because this is the #1 tumor suppressor gene, meaning that more than 50% of our human cancer has gene mutation or deletion on this particular tumor suppressor gene. This tumor suppressor gene to control the cell cycle proliferation by inducing P21 expression, okay? P21 and P53. Remember that because we're going to talk about in the next lecture as well. We'll talk about the P53 signal. P27 usually induced by differentiation process because of the cell committed differentiation. The first thing they do is withdraw from the cell cycle. So in order to withdraw from cell cycle need to be stopped. And in order to stop the cell cycle, they induce P27 gene for that purpose. TGF beta also have a role to induce P57. There's another protein called 1433 sigma, very